So if we do the blood pathway, it's too bad we don't have yellow markers.
It's cold in here.
Okay, if we had that diagram, we could label it, right? We start with the path of blood. So, good, Alex, what's A? No. I'm in the middle of it, I'm in the cortex of the kidney already, so I'm microscopic.
Wait, vocabulary, radiate, bone, something.
Half right.
Morgan saved his butt.
It's the apparent, capillary.
Arterial.
Oh, there it is.
Oh, sorry.
Right, it's the afferent arteriole going into what's, Alex, what's B? Shh.
Capillary bed,
It is a capillary bed, what's the capillary bed called?
whatever.
Yeah. Glomerulus, what kind of capillaries are they?
Fenestrated.
Fenestrated capillaries, okay, so we're going into C. Danielle, what's C? Efferent arteriole, don't forget the arteriole again, that takes us into D. Nifa, what have we got for D? He's wrapped around the tubule, so we're gonna call him no, we're not talking, we're in the blood vessels. Look at your blood vessel list. What are we gonna call a capillary that wraps around a tubule? A peritubular capillary. Okay, you got that one? All right, and then when the peritubular capillaries are finished, we exit out into E. What blood vessel? Alex? Hmm? It is not the renal. Get
No,
off the
radiate.
renal. Oh, radiate, okay. Okay, so that's the path of blood. Now, then we've got the path of urine. It's filtrate until it hits K. But let's see what happens then. Heather, what's that, F? Go. Anybody in the first row claim.
Bowman's capsule?
He's the Bowman's capsule. Both the person next to you.
Wait, what? Say that.
Bowman's capsule.
For which one?
Yeah, we're just gonna pass all the way down.
Proximal convoluted tubule.
Then we got G is gonna be a proximal convoluted tubule. H.
So loop of Henley?
Which part?
It's descending.
Yep, we're gonna be the descending arm of the loop of Henley, and then I is going to be
The ascending.
yay team. Hunter, what's J?
Oh, J is the distal convoluted tubule.
Okay, and that leaves us with K.
Collecting duct.
Voila. And you're a filtrate until you got to the collecting duct, because all the way through your contents are being manipulated. So, Mackenzie, first stage in urine formation is what? Oh. Okay, we got a filtration. Where is filtration happening, Anifa? Good question.
Glomerulus to the Bowman's capsule.
What is the problem with the glomerulus? Glomerulus. Okay, here we go. Somebody was sick in nursing
Lucky
today.
urine.
Okay, right, we're going from the blood into the urine, so this is filtration.
When you say blood, you mean bloodstream, right?
We're going from the bloodstream, we're going into the urine flow. Right, so this is filtration. Filtration is throwing out. Remember, filtration requires a high blood pressure, which is why these two guys have to be arterioles, so that we can change the diameter and change and maintain a constant filtration rate going on in there. Okay, so we've got the Bowman's capsule is filtrate. What's the next step?
Reabsorption.
We got a reabsorption. Where's that gonna happen?
Urine to blood.
Hmm?
Urine.
Urine to blood. Proximal
Okay.
convoluted tubule to Paratubular capillaries.
Reabsorption. We're going from the urine and we're gonna leave, we're going out into the bloodstream. Why? Why do we need to reabsorb? Oh, that's a good question.
Decrease your volume.
Because
We have to decrease the urine volume because we pulled too much water out. What else did we pull out by, accident?
We, amino acids and glucose.
Our nutrients are glucose and amino acids. Notice nobody wants to talk about fats.
Wasn't one of the
No, no, it
things.
isn't. It isn't on there kind of thing. But our nutrients that we just finished absorbing through the digestive system, now we just urinated them out and we've got to pull them back. What's the process for pulling them back? We got a sodium cotransport action. Sodium you guys are gonna have a long exam if you're gonna congratulate yourselves on the right answer. Okay.
Hey, not ready, so hey.
Sodium cotransport pulls three things back into the bloodstream. What are the three things that sodium cotransport is gonna pull back into the bloodstream? I think I might have counted four.
Protein.
It's not gonna be the protein. What's it gonna be?
Waste, glucose, amino acids,
Well, no, no,
urine.
hang on, no. Sodium cotransport, the mechanism, is gonna bring, you're right, if we're talking about an amino acid, whatever the nutrient is, we bring that back.
I guess I was talking about something.
Yeah, okay, so we're bringing back the nutrient.
Sodium,
We're bringing back sodium. If we bring back sodium, what comes back?
Oh,
water, electrolytes.
Chloride, okay, chloride ions. And then thirdly, water. So I've got nutrient, I've got sodium, I've got chloride, and I've got water. All of that's happening simply by sodium chloride. That's gonna pull back about 65% of the water. So it solves the problem of having too much urine. It solves the problem of bringing the nutrients that we filtered out by accident, kind of thing. But since we also by accident and using bulk flow bring waste products back into the bloodstream, we have to do one more step.
Secretion.
We're gonna do secretion. And secretion is going to happen where we want to throw things out of the bloodstream back into. This is also where we're going to have hormones. Hormones like aldosterone. Hormones like
A&M.
A and F. I'm trying to think. I don't want to say ADH.
You don't?
Nope, not yet. No, don't erase it.
Okay.
It's just not happening right there.
Okay, because I wrote it down.
It's gotta be another one. Oh, yes. The calcium one.
Parathyroid.
Parathyroid hormone. All of these guys are working in this particular area. So this is where the hormones can fine tune what stays in the blood and what comes out. What isn't affected by a hormone but is important as far as regulation again is also hydrogen ions. Okay, so I thought can we trace the path of each of those kinds of molecules through this diagram?
what?
Can we trace the path of each one of those kinds of molecules through the diagram? So, for example, what is going to happen to a red blood cell that's in the glomerulus? We got a choice. We can do filtration, reabsorption, secretion, all of the above, some of the above, none of the above. What a great multiple choice question.
Doesn't the red blood cell just go to the efferent and then to the paratubular? No.
Go ahead.
And then go out the radiate vein?
Right, so red blood cells should not go into the urine system at all, so there's none of the steps. Red blood cells are too big to be filtered unless the person has an infection, an inflammation of some sort. No red blood cells in urine, okay? What about creatinine? We haven't really talked too much about it. But we measured creatinine levels. It's an important level because what's gonna happen with creatinine is it's going to undergo filtration. only. So we'll put an X there because nothing happens. Creatinine gets filtered and then it stays in the urine. So when you do a urine sample on a patient, you're basically testing the filtration ability of the person's kidney. You know that nothing else is gonna mess with it. Creatinine test is gonna tell you how well the kidneys are filtering blood. Okay? Glucose. Somebody help me with glucose. What happens to glucose? We're gonna filter it, and then we have to reabsorb it because, oops, we didn't wanna throw it out.
And if there's excess?
Then
If
we
you-
better
if it's-
stop.
Huh?
And then we better stop. With glucose, we're going to filter it, but then we better reabsorb it because we wanna get it back into the bloodstream. The absorption capacity, the carrying capacity for reabsorption is measured as what? What's the limit? Yeah, what is it? The T-max, the transportation maximum, is the number of carriers that are available at any given time for absorbing. Okay? Urea, trick question. Okay, urea. What's the pathway for urea? It's a nitrogen waste, so of course we want to filter it.
You wanna secrete it.
Well, hang on, we wanna filter it because we wanna get it into the urine.
Okay.
But what happens because of bulk flow? Urea is going to get filtered out, that's a good thing, but then it's gonna follow everybody, it's gonna follow the crowd, so it's gonna get reabsorbed by accident. So then we still have to throw it out, so how are we gonna get rid of it again?
Secreted.
Secretion. So urea is going to do all of this. Okay, does that make sense?
Yes.
Okay, when I get to here, remember the diagram of a kidney, we had all of those collecting tubules coming together to form pyramids, then they go out into the pelvis and out of the kidney. That's what we're seeing here, is we're seeing the collecting duct, which is all gonna join together in the area of the medulla and then drain out of the kidney. So these can actually get almost macroscopic. But if we were stand right here, we would find that we've only reabsorbed 65% of the water that we need to reabsorb. So the last thing we have to do, if you've looked at all of that, we go back over all the list of the urinary system, been there, done it all, except we haven't balanced water volume. We haven't dealt with dehydration. And what that's gonna do is it's going to involve and this is why it's important, in the descending, have you looked at these kidney models in lab? Be careful, because remember the loop of Henle is flipped over on that one model, kind of thing. Okay, but in the descending loop, nothing exciting happens, sodium gets pulled out, chloride gets pulled out, and water follows. Nothing new. But when the fluid gets into the ascending loop of Henle, something strange happens. The sodium comes out, the chloride comes out, but it doesn't absorb water. Okay, water can't be reabsorbed at this point. So what's going to happen is we're going to end up pulling out a lot of sodium and chloride, but there isn't going to be any dilution of it. So we create a super salty environment between the loop of Henle, the distal convoluted tubule, and the collecting duct. Okay? That in of itself isn't terribly exciting, except remember the hypothalamus is measuring dehydration. When it senses that we're not retaining enough water, the hypothalamus is going to send a message, ADH. What ADH is going to target very specifically is the distal convoluted tubule and the collecting duct. And it's going to tell the cells of the nephron in that area to create special proteins called aquaporins. And that's simply pores that specifically are shaped to pull water molecules back. So what I'm going to do is right here. In this collecting duct, I'm going to make little proteins. And these proteins are going to be doorways that only allow water to leave. Okay? Aquaporins create pores if we remember way back, we said that at the end of a hormone story, whether it was a protein or a steroid, we said that the hormone's action was to increase protein synthesis in the cell. Now we know exactly what's happening. ADH is going to the cells that make up the collecting duct and the distal convoluted tubule and telling those cells to make these proteins. These proteins are called aquaporins, and they migrate over to the cell membrane, and they pair up and they make little doorways that only allow water molecules to leave. The more ADH, the more aquaporins, the more water is reabsorbed back into the body tissues, and the less urine we produce. Which we sort of know, we knew the beginning and the end of the story, we just didn't know how it worked. Okay, but that's not good enough. If ADH worked over here, if ADH made aquaporins over here, it wouldn't make any difference because the dilution of the fluid here and the dilution of the fluid outside the tubule would be exactly the same. So water would look out the open door and say, why should I go there? There's no reason because the dilution is exactly the same. But look what we did over here. We made the environment outside the collecting duct super salty, so that once you open a door, the water comes zipping right out. So that's why it's important to create a super salty environment just outside the collecting duct. And then when you open these special door channels, the water will want to come out. Do it over here, nothing's gonna happen. Do it over here, the water wants to leave. Yeah.
Just say one more time, the
Sure.
part from, the dehydration.
Okay, remember?
The ADH. and then-
Right, the hypothalamus is sensing dehydration, he produces ADH, ADH is coming down through the bloodstream, and he's going to those two areas of the nephron. Right? And what he's gonna tell those cells, 'cause remember, this isn't really a single line, it's a row of cells. And he's telling those cells to make these special protein molecules in their cell membrane. And that's going to allow the water to move through and back out. And then once it's out in the body tissues, it gets picked up by the capillaries and everybody's happy. But look at what we can do. We can regulate our water absorption. We can regulate our urine production by depending on how much ADH and how many aquaporins we make. So if the body feels you're dehydrating, make more aquaporins, and therefore you retain more water and you lose less urine.
So the ADH tells the DHC to make the aquaporins?
Right, because remember, the very first week we said, steroid or protein, no matter what the hormone is, the bottom line is that a hormone goes to its target cell and tells the target cell to increase protein synthesis. Now we have an exact, complete story. In this case, the ADH goes to the nephron cells and tells them to make aquaporin proteins that poke holes in the cell membrane and are shaped just to encourage the water to move through. Okay? We all good? Voila, kidneys. Anybody ever eat kidneys?
What?
Kidneys, anybody ever eat them?
Are you?
Unfortunately, yes. Usually the Thursday night of payday.
Liver.
Right. Kidneys and liver, that's what we used to have. I will never eat another kidney voluntarily. Okay, along with the chapter on the urinary system, right behind it is a chapter called Fluid and Electrolyte. And nursing tells me this is the hardest concept for their students to deal with. Not sure why. We won't suspect that it may be the way they teach it. Okay. The only thing new in this chapter is some categorization. We can divide fluids into intracellular fluids. And that's clinically abbreviated ICF. It's the fluid that is inside the cells, as compared to extracellular fluids. Cleverly, ECFs. The only fluid inside a cell is cytoplasm. But there's lots of kinds of fluids outside a cell. They're all pretty much based on blood plasma. But remember, from blood plasma, we can make cerebrospinal fluid, we can make lymph, we can make synovial fluid. We can keep going on and on. Okay, so blood plasma and all the other fluids that are outside a particular structure. I had a number. Okay. Of the two fluids, which do we have more of? Intracellular or extracellular? I would have guessed that too, but it's intra. 65% of your body fluids are inside cells. I mean, that's not anything I'm going to test you on, but I just thought that was weird because it sounds counterintuitive.
What's CSF stand for?
CSF is cerebrospinal fluid. Who did you have for A&P1?
Cool. It's kind of correct.
No, I don't know how to diplomatically do this, but I do have to find out about adjuncts, and nobody tells me until it's too late sometimes. But anyway. Okay, what else did we, okay. If we look at these two groups of fluids, the dominant positive ion inside a cell is going to be potassium. The dominant ion outside of a cell is going to be sodium. The dominant negative ion inside a cell is going to be phosphate.
What was the first one? The dominant?
That's potassium. These are the dominant positive ions and the dominant negative ions.
So potassium was inside, so.
This is inside. The dominant negative ion, chloride ions.
So
In
that
gen-
HP04 is negative?
is phosphate, it's two negatives.
Okay.
So it's a phosphate ion. This guy is generally going to have, it's going to be more viscous. It's going to have more proteins in it.
Which one, both?
I'm talking about the fluid, this column. Fluid inside the cell. Fluid outside the cell. So this guy's generally going to be more concentrated as far as protein goes. Other than that, we know everything else in the chapter. When we're talking about fluids, we're talking about water. Ooh. What do we know about water?
It's a shock absorber. It has surface tension because it is a polar molecule. Okay, polar molecule gives it the ability of attracting each other. Surface tension. Surface tension, in a sense, helps make it a shock absorber. It has volume. We think of that when we think about cerebrospinal fluid. It's what helps our brain float. Something basic. What's the formula? We got H2O. And remember, that means there's a big O with two little H's. So the oxygen pulls all of the electrons in one direction and therefore the rest of the molecule. So we end up with a little mini magnet. Helps with transportation of materials through various body fluids.
Regulates body temperature.
How so?
I don't know, it was a nutrition. It's one of her lists of things.
Water regulates body temperature. Well, it does in the sense that because it's a polar molecule, it doesn't change its physical form. So it doesn't freeze, doesn't crystallize, and it doesn't evaporate because it's a polar molecule. They like each other just the way they are. So we can say physically stable. Okay, as far as water is concerned, oh, I wrote that already. As far as water is concerned, the source is going to be our diet. How do we get rid of water? There's 3 ways at least that I can think of.
Sweat.
I got urine, you got sweat.
Wait, what is this? What are we talking
Evaporation.
We're talking about water. We're talking about the source of water is stuff that we eat. Let's not throw up. Let's be healthy.
Respiration.
Okay, so this is the source. This is how we get rid of it. We could also add fecal material in there. We could throw up if you wanted to, and stuff like that, but, okay. How do we regulate? Who's the hormone that regulates water in the body? Okay, if you guys have to think about this, okay, we're gonna regulate it using ADH. ADH comes from where? We're going to come out of the hypothalamus. The hypothalamus is going to send it over to the posterior pituitary, and the posterior pituitary is going to release the ADH. When does the hypothalamus? How does he know when to release ADH? When you're dehydrated, osmoreceptors. Osmoreceptors sense dehydration. So one of the things we're going to do, and you only have to have a 10% decrease in body fluid. Within 10%, your hypothalamus is screaming. It's going to do a couple of things, though, because dehydration threatens your blood pressure. And we know that the kidneys will shut down if they don't have enough blood pressure. So we have to take dehydration much more seriously than I think we tend to. So as soon as we sense dehydration, the hypothalamus is going to send out ADH, but what else is it going to do? AMP1.
It's
Going to make you feel thirsty so that you take behavioral actions. You're going to replace the fluid. Now, by the time the ADH gets down to the kidney and tells it to produce the aquaporins, blah, 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 kind of thing, you're well into being dehydrated. So one of the other names for ADH is what? Not suppressant, just vasopressin. And vasopressin is temporarily going to vasoconstrict. That keeps your blood pressure up while you're busy rehydrating. We need to, as soon as your hypothalamus detects dehydration, you've got to take immediate action, vasopressin. And then in the meantime, you're going to decrease your urine loss, and you're going to replace fluids in the body. So it's a 3-prong reaction. Dehydration is important.
Is that why they say when you're thirsty, you're already dehydrated?
Other than water, that's our fluid part. Then we have to worry about electrolytes. Electrolyte is simply a molecule that in water breaks down into ions. An electrolyte is simply a molecule like sodium chloride, salt. That when you add it to water, it breaks down into sodium and chloride. And the reason it's called an electrolyte is because any water that has electrolytes in it will conduct an electric current. Okay? If you're not an electrolyte, you're a non-electrolyte. They stayed up late thinking of that. And a non-electrolyte is a molecule that you can put in water and it will stay intact. Think fat. It stays fat in water. Think a globule of piece of meat type thing. So many of our organic molecules, they tend to be large, they tend to be structural. So we're talking about things like carbs, fats, proteins. They're all non-electrolytes. They're not what we're worried about. We're worried about them in the digestive system. And I only put this in because everybody talks about electrolytes, and I find I understand them better if I understand what's the alternative. So if I can mix it with water and it stays intact, it's a non-electrolyte. If I put it in water and it breaks up into elements, then it's what we have to worry about. The electrolytes that we need to know and love, most important, is going to be sodium. And we worry about sodium in the body because sodium attracts water. Sodium attracts chloride, which attracts water. So we always worry about how much sodium is in a cell, how much sodium is in a tissue, how much sodium is in a nutrient, because it's going to attract water. Water is going to affect your blood pressure. Water is going to affect edema. So we have to watch sodium. We're going to get sodium out of food. That's no excitement there. How do we hormonally control sodium? I can cry in public. So, tell me about aldosterone. Where did it come from? No. Where does source, target, action, stimulus? Let's start with where does it come from? Oh, you guys are going to have so much fun on a final. Comes from the adrenal cortex. How does the adrenal cortex know when to release it? What?
Increase of blood angiotensin.
Okay. We're going to start way back here in a kidney. Kidney senses I don't have enough sodium, or it senses a drop in blood pressure. I'm going to release renin. Renin is going to go out and tell angiotensinogen to become angiotensin. And when I see a lot of angiotensin, the adrenal cortex will react and produce aldosterone. Ooh. Where is angiotensinogen made? Where are many proteins made that run around in the bloodstream, the liver? Something well. Angiotensinogens, a classic example of one of those blood proteins that's made in the liver, sits in the bloodstream, not doing anything until it's called on. Okay? And when it just sits in the liver, it acts as part of the osmolarity concentration. Okay, so we got angiotensin, cortex, aldosterone. Now, where's it going? Where's aldosterone going to go? Better go to the kidney. Specifically, where in the kidney? Nope. Distal convoluted tubule. And he's going to tell the distal convoluted tubule to reabsorb sodium. If we reabsorb sodium, we reabsorb chloride, which means we reabsorb water, which means we increase blood volume, which means we increase blood pressure. Right?
Say kidney what, distal?
Convoluted tubule, DCT. Hopefully a couple of little gray cells are reawakening. We've done this. There is almost nothing new on the board. Okay. Well, that's if we want to save sodium. How do we get rid of sodium? Who's the antagonist? Not ADH. A and F, A and P, atrial natriuretic factor or protein. Oh gosh, now, if I'm gonna call him atrial natriuretic whatever, what's his source? He's coming from the heart. When is the heart going to yell for help?
It would be a work too hard.
Too much blood pressure. So I'm going to react by producing A and F. Where's A and F gonna go? It's gonna go to the kidney. What part of the kidney do most hormones target? And what's he gonna tell those kidney tubules to do? He's gonna tell them to pull sodium out of the bloodstream. We're gonna secrete, we're gonna lose the sodium. And if we lose the sodium, we lose the chloride, we lose the water, we lose the blood volume, we lose the blood pressure, which solves the problem. What do we call a reaction where we have a stimulus in one direction and a reaction in the opposite direction? Negative feedback. Negative feedback, that's how most of your hormones work. If you know one end of the story, you can figure out what the other end of the story is. Okay, that's sodium. The other important ion, and we can call them ions because that's what electrolytes become. The other important ion that we have to follow is calcium. Yay, calcium! We haven't talked about calcium in ages. Okay? So we have calcium. Why do we care about calcium? What about the heart regulation? All muscle contraction. All muscle contraction. So we need to keep calcium at just the right levels for muscle contraction. Too much calcium, you become irritable or twitchy, which is not good for a heart. Not enough calcium, you become sluggish, which is also not good for a heart. And I did something terribly dastardly to the A&P1 class. I asked them about neuromuscular and muscle contraction, and I asked them about calcitonin. And they all read the question as calcium. So we all got it wrong. But we'll get back to that. Okay, so we need it for muscle. What else do we need calcium for? That's an Acme answer. What about this semester? What about prothrombin? Clotting. Remember all of the vertical lines in those clotting diagrams, calcium is required. You don't have enough calcium, slower clotting rate. Too much calcium, you're facilitating intrinsic clotting. So we can worry about clotting. We can talk about that neuromuscular junction. Because remember, acetylcholine doesn't get released unless calcium tells it to. We also used calcium as a second messenger. sure we all remember, it's right in your frontal lobe, I can see it. Remember that protein hormones can't get into the cell. They have to use a second messenger to go into the cell and tell the cell what to do. Calcium can be a second messenger. So again, calcium is awfully important for lots of different reasons. So then the question is, how do we regulate calcium? Too much calcium, what hormone is going to be produced? Who's going to produce the calcitonin? We have too much calcium in the bloodstream, we're going to produce calcitonin, but where does it come from? We're going to get it from the thyroid. Thyroid's going to produce calcitonin. What's calcitonin going to do? Calcitonin in. He's going to go to the bones and tell them to do what? Because what's our problem?
Release calcium.
Pull it in.
Pull it in.
Okay? We're going to go to the bones and tell the bones to increase storage. And that lowers our blood calcium, which solves our problem. Okay? Another negative feedback. So when I ask the class, if you don't have enough calcitonin, what's going to happen? If I don't have enough calcitonin, I'm going to have an excess of calcium in the bloodstream. And so the muscles are going to be more irritable, because they read it as calcium, not enough calcium, and they answered all the other way. Oh, well. Mispopularity here. Okay, what happens when we don't have enough calcium? Who cares, and what do they do about it?
Parathyroid gland releases parathyroid hormone.
He's going to release parathyroid hormone, and what's parathyroid hormone going to do? No, it's not going to heavily sigh. Go ahead. Well, first, take the easy one. Right? He's going to go to the bones. He's going to tell the bones to release calcium. So it's a bone thinner. Okay, so I'm going to take it as storage. What else am I going to do?
Go to the kidneys and tell them.
Yay, team, we're going to go to the kidneys. What part of the kidneys, by the way? The distal convoluted tubules, because that's the part of the nephron that hormones work on. So we're going to go to the kidney. We're going to go to the distal convoluted tubule, and we're going to tell the kidney to retain or reabsorb. We'll use our urine words. We're going to reabsorb calcium from the urine.
Is that just in the urinary system that we do that? Like go to the distal convoluted tubule?
At any given time, you're going to go, parathyroid will go to 3 places. And one of them is going to be the kidneys. Because you're always making urine. 180 liters of it, last I heard. 3rd place. Supposing you've been chewing on bones. Oh, I'm sorry, that's finals week. Trying to think. What's the 3rd place we're going to do to look for calcium? I've looked in the bones. I've looked in the urine.
Small intestine.
Small intestines. I'm going to go to the small intestines, and I'm going to tell them to, again, increase the reabsorption. All of those together should bring our calcium levels back up. We've solved the problem. So the electrolytes that we have to worry about: sodium, calcium. The 3rd one is hydrogen ions. Hydrogen is the last of the electrolyte, or ions really, that we need to work